like first to say thank you to the organizers. I'm really thrilled to be here. It has been a great meeting. And yes, I'm going to talk about neurophysiology of rhythmic timing in macaques. So, wow, what's going on? Sorry about this. So I'm just going to start with a definition besides beat-based timing. Beat-based timing refers to the ability to detect regular pulse or beat in music and to respond synchronously to the beat during dancing and musical ensemble playing. So this is a highly complex cognitive ability. What I'm showing here is a score of music and the corresponding auditory input. So the human brain is, very, very capable of extracting the beat structure of this auditory input and to generate a pulse internal representation that is isochronous and regular. And this internal representation doesn't necessarily follow all the properties of the auditory input. It's a cognitive construction of a beat that is regular. Once you extract the beat, something that happens normally is that you have the urge to move, with this beat. And in the lab, what we do is to measure normally tapping in synchrony with this stimuli. And this tapping has two main properties. One of them is that it occurs a few milliseconds before the internal beat. And second is a very nice measure of the internal representation of the beat. So different studies that measure EEG in humans have found that the tapping times are really related with this internal representation of the beat. Doesn't matter what is the internal, the auditory input. So this complex behavior that has, bottom-up and top-down components is one of the main research goals in my lab. And we have developed a research program to try to understand what are the neural correlates of an internal clock. And then for that, we need a model. So for a long time, using behavioral and EEG measures, we have demonstrated that monkeys have the ability to perceive and flexibly synchronize to a metronomic beat, but also to beats that are more complex, like simple integer ratios, like the one that Andrea Ravigiani presented yesterday. So 10 years ago, in fact, with Hengyang Honing from the University of Amsterdam, we suggested that beat perception and entrainment have emerged gradually in the primate order. The large human flexibility to entrain to complex music beats depends on the close dynamic interaction between the auditory and the motor system. However, monkeys have all the neural machinery to perceive and entrain to simple beats, such as a regular metronome, since it has a complex auditory cortex with a posterior stream that connects to the posterior parietal cortex, and then there are projections to the medial premotor areas that are connected with basal ganglia generating an internal clock. So how do we measure these in the lab? We train monkeys in tapping tasks, and the classical one is a synchronization continuation task. Professor Palmer already talked about this. This is a monkey version of it. So we have a metronome defined by brief stimuli with regular time between them, and the monkey is trained to tap in synchrony to this stimulus for 3 intervals and then continue tapping for other 3 intervals without the advantage of a sensory input. In this case, the monkeys were trained to produce intervals that were below a certain error. And if that was the case, they receive a reward. And we keep very dynamic, this stack very dynamic by changing the duration of the metronome using 5 different durations. In this case, we train monkeys to do these tapping tasks using visual or auditory metronomes. And here is an example for the auditory condition. I'm going to play it again. Well, if I can. So you see that the monkey has a very stereotypic movement, and it can time, the metronome by pausing the movement between the stimuli. So we have characterized what were the properties of the behavior of the monkey using auditory and visual metronomes, and compare that with humans. This is something that we do a lot in my lab. We use exactly the same task, the same hardware, the same software, and then we compare the 2 species. In this case, this is a measure of accuracy. It's the constant error, that is the subtraction of the produced minus the target interval. And you can see that the monkeys produce shorter intervals than humans, but nevertheless, they have a pretty nice performance. Monkeys also show the scalar property of interval timing, which means that the standard deviation of the produced intervals increases a function of the time interval, which is, a very common type of behavior in timing tasks across species, including humans. So once we trained monkeys and we know that they were able to do the task, we first were wondering where to record to try to understand what are the neural correlates of an internal beat clock. And a long time ago now, with Warren Meck and Deborah Harrington, we suggested a relatively simple hypothesis of how the brain can time things. We suggested that timing depends on 2 components. One of them is the main core timing network that includes the medial premotor areas, the basal ganglia, and the motor thalamus. And then a series of context-specific structures that interact with the main core timing network to define the timing behavior in a specific context. And this is important because timing is not homogeneous. The exactitude and the precision of timing depends a lot on the context of the timing. And during years, we have been focusing on the neural properties of these areas, the medial premotor areas that include pre-SMA and SMA, but also we have recorded other areas. In this case, however, I'm going to focus mainly on the medial premotor cortex. So something that we did just at the beginning is to try to understand how were the changes in instantaneous activity during our task. And we found different types of ramping activity. One of them that was pretty common were classified as motor cells. So these cells increase their firing rate just before the tap. And this activation profile was very similar independently of the produced durations by the monkey. However, we also found neurons before the tap that can be measuring how much time is left for the next tap. So these are time to contact neurons, and their property is that they have a duration that scales with the produced interval, and they have shorter slopes for shorter durations, which produces that these neurons reach a particular peak, at a particular time before an event. And this type of, predictive type of signals have been reported in many other timing tasks. We also found neural correlates for the quantification of elapsed time. And we found 2 types of ramping activity. Some of them were up and down, and that scale in time shrinking and elongating depending on the produced interval, which are what we call here absolute timing cells, and cells that look like some sort of SyNC clock of neural activity. These neurons reach a peak that is related with the time duration. So we found this ramping activity, but looking carefully to the signal, we also find tuning. Already Dean Buonomano showed that there are many principles in recurrent neural networks that allow you to generate tuning for different intervals. What we found is that neurons could be tuned to shorter intervals, to longer intervals, but also were multiplexing serial order. So these neurons then could respond in a particular element in the sequence of rhythmic movements. So in this case, it's the fourth element of the sequence, and is, has a preferred duration for shorter intervals, and this other neuron has a preferred duration for longer intervals, but is responding at a different serial order element. So you already have two components here. Ramping activity on one side, that is quantifying elapsed time and also predicting the time remaining for an action, and then you have neurons that are tuned to intervals and serial order. So the picture became a little bit more complex when we saw the temporal profile of activation of these neurons. So these are the serial order neurons, the neurons that are related with serial order. In this case, the x-axis is the 3 synchronization intervals and then the 3 continuation intervals. And on the y-axis, you have the cells, and the color represent neural activity. This neural activity was sorted for the peaks, and what you can see is a pattern, a progressive pattern of activation for each of the produced intervals in both type of populations. So it means that this clock is a complex clock. It has tuning components, it has ramping components, and also it has neural sequence components. So to try to understand how this is represented dynamically, what we did is to use dimensional reduction techniques. These techniques have the goal to determine the shared pattern of activation in neural population based on the response covariance as captured by latent variables like the PCAs. Then what we do is to build a state space diagram by plotting the first principal component against each other. And then what you get is, in every moment of time, a state of the population that is shared across the neurons that we recorded based on a reduction, dimensional reduction technique. There are many reductional dimension techniques. We have used many of them. In general, the type of properties that we get, are independent on the technique. And what I'm showing here is just the neural trajectories when we project these PCAs using 3 dimensions or 2 dimensions. And here in the corner, we have the monkey doing the task. So what we found in this case, this is using 1,500 neurons during task performance. We found this type of neural trajectories. What's going on? I don't know what's going on.
Get rid of the laser pointer probably.
Okay. Here we go. So what you see is, the neural trajectory starts at a particular, point in state space, and then they reach a particular orbit in task performance, and what they do is that they rotate for every produced interval. Something that is important is that this rotation is similar across produced interval in a sequence, but the tapping times occur at a particular space within these trajectories. We found also that the amplitude of these circular trajectories increased as a function of the duration of the time interval. So we have larger circles for longer durations. And we found this for the synchronization and the continuation epochs of our task. In fact, when we measured the radius as a function of the target interval, we found a monotonic function. Another property of these neural trajectories is that they form what we call tangent circles. They tend to have some sort of attractor behavior in a particular part of the circular trajectory, and then for longer intervals you have larger amplitudes, but the tapping times occur at a specific state space portion. And then to try to quantify these parametrically, what we did is to compute the Euclidean distance between an arbitrary point and the tapping times, and also this distance for half the inter-tap interval. And what you can see is that this distance is constant for the tapping times, suggesting that there is some sort of attractor behavior during the movement, and during the duel between movements, there are changes in amplitude of this trajectory. We also found that the variability of these neural trajectories increases as a function of target interval. And this could be then a neural correlate for the scalar property of interval timing. The trajectories that are more variable are related with intervals produced in a more variable way because they are longer. So I'll summarize some of these. Neural activity show a strong periodic pattern when projected to a lower dimensional state space. Each produced interval is represented as a periodic loop in the neural trajectory, conforming a regenerating pattern of bit-based temporal predictions. Neural trajectories converge to a similar state space at tapping times, resetting the bit-based clock at this point. And then the scalar property, that is a hallmark of timing behaviors, was counted by the variability in the curvilinear radia of the neural trajectories. So the next step was to compare what was going on between modalities. And then in this particular project, what we did is to train monkeys to just short and long intervals, and we compare the behavior and the neural activity when we use auditory or visual metronomes. In this case, it's important to emphasize that the task had a perception epoch where the monkey was not moving, was just waiting for a go signal, and once that signal occurred, they were synchronizing. And something that is important in this case is that the reward contingencies were more emphasizing prediction. So the monkey received a reward only when the timing between the tap and the stimulus was relatively short. So here is another video of a monkey synchronizing to this. So you can see that the timing of the monkey is pretty good, and it can switch from interval to interval in a very dynamic fashion. They can do this not only for isochrony, they can do this for short, long type of beats. So as I mentioned, monkeys have a bias towards visual metronomes, which is the contrary of what we found in humans. So this is the variability, which is the standard deviation of the produced intervals for the short and long intervals. You can see in blue that the variability for the auditory modality is quite larger than in the case of visual. And if you measure the accuracy, again, the monkeys can do this pretty well, but they are more variable for the auditory condition. So something that we did to try to understand well what was the behavior behind how the monkeys solved the task is to measure the speed of the tapping. So here you have the speed computed from video analysis of the position as a function of time. And what we found is that the down-up type of movement of the monkeys were very stereotypic. They were composed of two Gaussian speed profiles that were very regular across the sequence. This dot here is the tapping time. And if you use a threshold, you can divide then the movement times from the dwell times. And you can see here is that the movement time is very regular across conditions, but the dwell is longer for longer tempos. So what the monkeys are doing here to solve the task then is to generate a stereotypic movement that is probably controlled by some sort of pre-stated motor program. They tap at a particular time, and this probably is related with the internal beat, as has been seen in humans. And they really solve the timing task by timing the dwell. So we have three processes, and we were interested in trying to find what were the neural correlate of the three type of processes. So for that, what we did is to use these silicon probe arrays that allows us to record almost 100 neurons simultaneously while the monkey is performing the task. And what I show here are the neural sequences associated with the auditory and visual conditions for short and long. Again, you have time in the x-axis where each black line represents the tapping times. And then you have the cells in the x-axis. You can see clearly neural sequences building up across all the tasks with some resetting. So the first thing that we did is to try to understand what were the neural trajectories associated with these conditions, and what were the geometrical and kinematic properties of these neural trajectories. So these are the neural trajectories for the three first PCAs for our 4 task conditions. You can see again many things that we already saw in previous experiments, which means each loop is associated with each produced interval. The tapping times are in a particular part of the state space. There are changes in amplitude as a function of the duration of the rhythmic timing. And you can also see that there is an effect of modality. So you can see that auditory and visual trajectories occupy different positions within the state space. So the first thing that we did is to try to determine what were the coding dimensions of these neural trajectories. And we found a duration subspace plane where you can see clearly that the trajectories were had a larger radius for longer durations in both modalities. We also found some, a linear manifold that is related with the tapping times within this state space. And something that is interesting is that this line is pretty much orthogonal to the duration plane, suggesting that you have populations of neurons related with the tapping and the movement versus the ones that are timing the dwell between movements. We also found, the modality subspaces that had a particular angle between each other. This angle was around 59 degrees, suggesting that what we have here is shared resources, but they are not totally overlapping for visual and auditory metronomes. And we also find the serial order subspace, which has to do with the sequence. This space for this particular task was not particularly important. The variance explained by this serial order and subspace was small. Then what we did is to see if there was some sort of general timing mechanism we found, that these neural trajectories could be, summarized by this type of a circular clock that is an amodal representation of beat-based timing, and it has two components. The trajectories converge in a similar state space at tapping times, providing a phasic representation of a cyclic time event. And then you have this continuous and relative measure of how much time has passed within each produced interval. So we have a relative timer here that is telling you what's the proportion of time that has elapsed. But we also found neural correlates for dual processing. And we found, in fact, two kinematic properties for the neural trajectories that were related with counting or timing the dual. The first one was the amplitude of these neural trajectories. If you quantify this amplitude during the dual, you can see a clear increase in amplitude, which is not the case for this same amplitude during the movement period. So this is a dual-related property. So we also saw that the modality, what it is to project these circular neural trajectories in different spaces within a representation, but all the properties of these neural trajectories were very similar between the auditory and visual conditions. And this is suggesting that we have here a clock that has an external input from the medial premotor areas that is generating this switch in state space. What I show here is that the variability of these trajectories in this plane increases, as a function of the interval, and was more variable for the auditory and the visual condition. So this means that probably these neural trajectories are really related with the behavior that we are computing. Another thing that we saw is that the speed of the neural trajectories were also modulated by the dual. Okay, so if we quantify the speed during the dual, you see faster speeds for shorter intervals, suggesting then that you have temporal scaling in the neural trajectories in addition to the amplitude modulation. And that's quantified here. This is the speed for the movement, and this is the speed for the dual with larger speed for shorter intervals. So since we have a competition between amplitude and speed, we develop what's called an amplitude modulation timing scaling index that has values of -1 when you have only temporal scaling, and has values of 1 when you have only amplitude modulation. And what we saw is that there are cyclic patterns of these two indexes, for the amplitude and the velocity, but the conjunction of the two were close to 0, suggesting, a close relationship between the two parameters during dual. When we also focus on the movement times, and we compared, in this case, we correlated the speed profile of the trajectories against the speed profile of the hand movement, we saw clear correlations. Of course, the speed of the trajectories have changes that occur before the changes in hand speed. So this is, again, a narrow correlated, but for the movement. So we can say that there is an amodal representation of beat-based timing with two components. The trajectory converges at a similar state space in tapping time. And then you have the continuous lecture of the produced interval. We can say that the tempo of the tapping synchronization depends on the dual between stereotyped movements, and this duel is encoded by a combination of amplitude modulation and temporal scaling. And the modality imprints specific signatures in neural trajectories with a large displacement in state space without greatly altering the cyclical organization, duration-dependent changes in amplitude and temporal scaling, nor the tap attractor behavior. So in general, this can be interpreted as rhythmic timing to metronomes with different modalities depend on the interaction of a modal internal representation within the medial premotor areas and a modality-specific external input. At least this is one of the hypotheses that we are handling now. So the next step was to try to see if there are things in the neural sequences that are related with the properties of the neural trajectories that I just shown. So the first thing that we did is just to do simple, regression and mutual information analysis on single cells, and we found mixed selectivity for duration, modality, and serial order, and all these cells, had a pretty strong component to compute elapsed time too. You can see that the representation in single neurons is really distributed across all our parameters with high mutual information for duration, modality, serial order, and elapsed time. And most of the cells encoded more than one parameter. So we're talking about a complex type of population, type of responses. And here I'm showing some profiles of activation. So for example, this is a neuron that is selective for the short auditory condition. This is a neuron that is selective for longer duration, no matter what is the modality. Here you have, a duration selectivity neuron. So, we have a quite large variety of neural responses here. So to try to quantify the properties of the neural sequences that Did Bonomano, was talking about yesterday, what we did is to detect the beginning and end of the activity profile of each neuron. And this is represented by a line here. So again, here you have the x-axis is time, here you have cell number, and then for each produced interval, you have a neural sequence. And then, in general, you can say that each of these neural sequences flexibly fill each beat duration depending on the tapping tempo, provide a relative representation of how far an interval had evolved, and an important and crucial thing is that they reset at each produced interval. So once we have this information, then we characterize which parameters were related with our independent parameters in the task. So the first thing is that we focus on the duration of the activation periods for each of the produced interval across our conditions. This is the mean for all the serial order elements. You can see that the duration of this activation period is longer for longer intervals. We also computed the recruiting lapse, that is, the timing at which these neurons are, recruited within a neural sequence. And you can see oscillatory patterns for across the sequence with a faster recruitment lapse for longer durations. And then we also computed how many neurons we have in each neural sequence. And then you can see that there are larger amount of neurons for longer durations. So all of these neurons were related with neural sequences that mainly occur within the tapping time. But we also found neurons, what we called before in the ramping study, neurons that were motor related. These neurons were activated before each tap, no matter what was the serial order element, no matter whether it was short or long, or we use auditory or visual cues. So these neurons then have a similar profile across serial order and task conditions, and probably they are related, with the process of triggering, the down-up tapping movement, which is stereotypic. So if we try to summarize what's the relation between the circular trajectory properties with the properties of the neural sequences, we found three main relations. The first one has to do with the tapping time that occur, between the down and up movements of the monkeys, and this control of a stereotypic movement, we believe that is related with the speed of the neural trajectory during, just before the movement. And this is related then with the activation of motor cells that has an activation profile that is covering the movement and even a little bit before. We also found then a neural correlate for the internal beat representation at tapping times. So in this case, the neural trajectories converge in a particular state space of these circular trajectories, and this is related with the resetting of the neural activity. Every time that you have a sequence that ends in this reset, we believe that this resetting process produces this pulse that is then distributed across the different circuits that are related with the behavior. And then the dwell, the neural correlates of dwell that are related with the amplitude and the speed of neural trajectories are related with two properties of the neural sequences. So for the amplitude, what we have is a larger number of neurons for longer intervals. And for the speed, we have changes in the duration of the activation period. So these are just simulation that allows you to have this type of properties. So it means that there is a tight relation between the neural sequence properties and the neural trajectories. So what's our hypothesis behind what we found? So if you have these complex neural trajectories that are dependent on the duration, the movement, the tapping time, and also the modality, so how can we frame these in a particular hypothesis? So we're using a dynamic system approach where we have two components. The first one is f of x that represents the interaction between cells within the clock, in this case the medial premotor areas. And then we have u that is associated with external inputs to the medial premotor cortex. And this u then, what it does is it displaces the trajectories in a different state space, but the properties are relatively similar and kept for the two modalities. So how do we represent this in anatomical ways? Our hypothesis is that this modal neural clock is not only in the medial premotor areas, it's also in the basal ganglia thalamus loop. But this U, which is the external input, probably depends on the very uneven density of projections from the parietal cortex to the medial premotor areas for the two modalities. In the case of monkeys, we know that the visual dorsal stream is really well connected to the posterior parietal areas. And these posterior parietal, they provide a very strong visual input to the clock. In contrast, the auditory input to this posterior parietal cortex is really small. It's there, but it's small. And then the hypothesis is that this difference in input magnitude is what generates an external input that provides these changes in state space projection for auditory and visual. So how I'm doing with time?
20 minutes. We have
Ah. So what I'm going to present now is things that are not published. And we are very interested not only in the clock, we are very interested also in how is the input of these metronomes processed in the auditory cortex or the visual cortex, how the information is also processed in this posterior parietal cortex, and how these areas are interacting. So to try to understand that, what we do is simultaneous recordings in many areas while the monkey is performing a task. So we recorded in the core auditory area and the posterior belt auditory areas. We recorded in the medial premotor cortex, also the primary motor cortex, and the putamen, which is the entrance of the basal ganglia sensory motor loop, and also in the posterior parietal cortex, specifically in area 7A, that has access to auditory and visual inputs, and also projects to the premotor system. So again, we used a synchronization task that has a perception epoch where the monkey is just waiting for a go signal, and then after the go signal, the monkey produces the taps. We also have some other conditions in which the monkey can continue without the metronome, which will be a synchronization continuation task. Or we can have omissions of the stimulus in particular places to see how the processing is related with the auditory or with the prediction. And this is the behavior. This is the behavior of two monkeys. What I'm showing here is the asynchronies, which means are the times between the stimulus and the taps in polar coordinates. So negative angles represent prediction, and positive angles means that the tap was a little bit after the stimulus. You can see that there is a bias, so more prediction for longer intervals and less prediction for shorter intervals. That's very similar to what Professor Palmer shows yesterday for humans. If we quantify, these timing relations in a task where instead of having isochrony for the stimulus, you have intervals that are randomly presented, so the monkey cannot predict, the monkey will react. You can see that clearly, these reaction times become longer. So the monkeys are predicting, and they are switching tempos in a very flexible fashion. So the first thing that we try to focus on is on the frequency domain in auditory responses. This is the LFP, fast Fourier analysis. Here you have just the change of voltage of the LFPs, and here you see the spectrograms. What I'm showing on the top is the activity in the auditory cortex when the monkey is listening to these metronomes, but is not doing anything with it. It's a passive situation, it will not receive reward. Here you have the active task where the monkey is tapping in synchrony, and here you have the difference. So something that is amazing here is you see quite stereotypic responses with short duration to each of the stimulus presented in a metronomic fashion for the perception and the synchronization epoch in blue, okay? And then here you have, this spectrogram for the different frequency bands. You can see for the case of beta, there is a humongous inhibition once the monkey starts to tap, here shown in red. And that you have these cyclic rebounds in the beta signal. But in the case of longer durations, you can see that there is a larger inhibition that it is somehow relaxed when the monkey is engaging in this synchronization. Something similar happens for the low frequency band, delta. So this is for the LFPs. What happens with the spikes? So what you have here is the population of neurons that responds to the metronome input. They have a very phasic response, a very short duration response, and here you have the principal component 1 for a longer duration and a shorter duration. You can see quite stereotypic type of responses. So, and here, this is a signal that is really related with the auditory input. There's not a clock here. However, it was amazing. I mean, this is something that really surprised me. We have a large population of neurons in auditory cortex that are showing a suppression for each of the stimulus. And then they have a rebound. Okay, so here, this is the perception epoch, and then here you have the synchronization epoch, and you can see how these neurons siling up, and then there is a rebound. So here you have a very complex type of picture, right? This is not a sensation only. And we are very interested in characterizing what's the balance between excitation and inhibition, and whether these type of rebound signals are related to building the first auditory clock. But then what we did is to use statistics to try to see whether the neural activity across areas is related with the metronome, with the sensory input, or is related with the tapping behavior. So what we use is an algorithm that we developed with Rob Cass from Carnegie Mellon University, that what it does is to align the neural activity to the sensory events with this constant at 0, this W, it's at 0, but when we warp the neural activity and we get alignments to the movement, then this constant becomes 1. And we can use likelihood functions to see whether the neural activity is better statistically aligned to the sensory, to the motor, or even to complex events with W values that are within, around 0.5. So of course, in auditory cortex, we found a lot of sensory neurons using this method. So these are neurons aligned to sounds. Of course, this is the passive situation. The monkey is not doing anything. Here is tapping with you have the perception and the synchronization epochs. But if this neural activity is aligned to the movements, you can see that really this activity is not well aligned to the motor events. And this is the population of sensory neurons for the core auditory cortex and the belt. You can see clearly this activity with a very sharp increase in activity and short latencies with respect to the stimulus. But in the same auditory cortex, we found neurons that are aligned to the tapping movements. And we believe that this neural activity is related with the top-down signal of this internal clock. So these neurons are much better aligned to the taps than to the stimuli. And if you see at the population level, you can see that this neural activation occurs just before and just after the tapping times. And this is again the same circuit. So it means that the auditory cortex is not only sensory, it has a lot of processes related with the task. So we compare the proportion of neurons that were classified as sensory, motor, and complex for the auditory, the belt, this is the posterior parietal cortex, PSMA, putamen, and the primary motor cortex. You can see a clear increasing gradient for motor signals in green, and a clear decrease in sensory signals in blue. And something that is interesting is the posterior parietal cortex has a lot of complex neurons. So it means that you have a mix between sensory and motor signals that probably are related with comparing the sensory representation and the internal representation of the beat. So another thing that we have done, and I will finish almost with this, is to try to understand whether the intrinsic time scales, which are, is a measure that was developed by Shangwing Wang, and is related with the autocorrelation of the activity during rest, during a control period. This intrinsic time scale constant is telling you how fast the system is processing information and whether the information is kept in the system if you have a longer tau, or is really a fast processing. It's a little bit related with the notion that Dean Bonomano was talking about yesterday. And what we see is that this tau is shorter for the auditory areas, and it's longer for areas that are higher in the processing hierarchy. So, of course, we need to do much more analysis to try to understand how these areas are processing, the sensory part, the internal part, the motor control part, and also the error correction between all these signals. In fact, I'm very interested in trying to find out what are the neural correlates of error correction. So what I have here is just a diagram of what are the possible 5 elements necessarily to produce audiomotor synchronization. I'm not going to take too much time, but it goes from the primary auditory cortex, from the posterior stream of auditory processing, posterior parietal cortex, the clock in the premotor basal ganglia loop, the primary motor cortex command to do the tapping, the stereotapic tapping. And we also believe that there is an important role of the substantia nigra pars compacta providing dopamine to the basal ganglia when the performance is reward related, which means when the monkey has asynchronous that are close to 0. So finishing, I would like to acknowledge the hard work of a lot of people in my lab, including Yaneri Ayala, Juan Pablo Marquez, Germán Mendoza, Jorge Gámez, Abraham Betancourt, Luis Prado, and Raúl Paulín, and also the collaboration of a lot of brilliant researchers across the globe. And I would like to say gracias por su atención.